Hello everyone, welcome to BioLessons. In today's lecture, we are going to talk about a very important NEET topic, that is, how is urine formed? So by the end of this presentation, you should have a clear understanding of what is urine, what is it composed of, what decides its composition, and what are the interesting facts about urine formation. So let's see what's inside the topic. Urine is formed when blood is filtered out in the kidney. Okay, now blood consists of different blood cells and plasma protein dispersed in the water medium. Along with that, we have glucose, amino acids, salts, which act as a useful nutrients. Also, we have urea, which is the nitrogenous waste. Now, what happens is in the kidney, the blood is filtered and this modified filtrate is known as urine. Typically, it consists of nitrogenous waste that are toxic to the body, such as urea, trace amount of uric acid, creatinine, and whatever salts are present in excess amount than that of which the body requires. All of these are present in the urine. Okay, normal urine, if you see normal urine sample, it should not contain any glucose in it or any amount of blood cells in it. The main site for urine formation are nephrons, that are the structural and functional unit of kidney. The formation of urine, it takes place in three main steps. The first one is ultrafiltration. Here the blood is filtered under very high pressure and it takes place in glomerulus. Second step is reabsorption. Useful substances that are present in the filtrate are returned to the blood and is known as tubular reabsorption. The third step is tubular secretion. Okay, additional waste that are present in the blood are given back to the filtrate and this is known as tubular secretion. Now this reabsorption and secretion that take place in the renal tubule. Clear? Let us now understand the first step of urine formation, that is ultrafiltration. This is the human kidney. This is the renal artery which carries in the impure blood to the kidney that has the nitrogenous waste in it. And this is the glomerulus. This is the afferent arteriole formed by the fine branching of the renal artery. And afferent arteriole carries in the blood to the glomerulus. This is the efferent arteriole which carries away the blood from the glomerulus. If you notice carefully, the afferent arteriole is wider in diameter than that of the efferent arteriole. Now, this difference in the width of the afferent and efferent arteriole creates a pressure difference. This high filtration pressure causes the blood to pass through the filtration barrier that has three layers. And this results in formation of the filtrate that is present in the Bowman's capsule. Okay, so what's inside the filtrate? All the small molecules can pass through, that are water, ammonia, glucose, ions such as sodium and potassium. All of these are present in the filtrate. And what cannot pass through? All the bigger molecules such as protein, blood cells, they cannot pass through the membrane and are retained in the blood. Okay. Let's now see in detail the filtration barrier. Pay close attention to the figure here. This is the afferent arteriole which is wider than the efferent arteriole in diameter. The filtration barrier is made up of three layers in it. Okay, if you see, the blood has to cross three layers. First, it has to cross the capillary network, then it has to cross the white space that is in between the capillary network and the Bowman's capsule. So that second layer is the basement membrane, that is thick and non-porous. And if you observe Bowman's capsule, this is its outer layer, this is the inner layer. The inner layer of the Bowman's capsule is made up of the visceral epithelium, and that acts as the third filtration barrier. Okay, let's look at this figure which will make it more clear. This is your capillary endothelium. Okay, this capillary endothelium is fenestrated. What is the meaning of fenestrated? It means it has small pores in it. These small pores will allow only the small molecules to pass through them. Okay, and larger molecules such as the blood cell and plasma proteins will be retained here. Okay, the second layer is the basement membrane, that is thick and non-porous, and the third layer is the visceral epithelium of the inner layer of the Bowman's capsule. Interesting point is this visceral epithelium is made up of podocytes, okay, which are projections, and these act as the filtration slits or slit pores. Okay, this is the final layer which the blood has to cross and the filtrate will be present in between the two layers of the Bowman's capsule in this area. Okay, so as a result of this filtration barriers, what happens? The filtrate that is present inside the Bowman's capsule has only the small molecules in them, and the blood retains all the large molecules such as the blood cell and the plasma proteins. Is that clear? Let us now understand some important terms. Glomerular filtration rate or GFR. It shows how much volume of filtrate is produced by the kidneys per minute. It is a measure of how nicely the kidneys are functioning, okay, or the nephrons are working. The normal GFR is found to be 125 milliliter per minute. So the filtrate that is produced per day is about 180 liters per day. Okay, but you see, humans are land animals and we cannot afford to lose so much water. So water is reabsorbed by the body with the help of the renal tubules, and the final urine output is just about 1.5 liters per day. Okay, let's see that more in a little bit detail. In the last slide, we looked into the first step of urine formation, that is ultrafiltration, the filtrate present in the Bowman's capsule. Okay, now we have three major challenges with the filtrate. Number one, in the filtrate there is too much of water and we cannot afford losing that much water. Second, it has essential nutrients which we again cannot lose in the urine. Third, the blood coming out of the glomerulus still has some nitrogenous waste as well as H plus ions that needs to be taken out from it, otherwise it would be toxic for the body. So what happens, the peritubular capillaries that are close to the renal tubule, they help in achieving the uptake of the essential nutrients from the filtrate through the process of tubular reabsorption. That is a second step in the urine formation. Also, what happens, the waste present in the blood is given back to the filtrate, and this is the third process of urine formation, that is tubular secretion. Okay, let us now look how specific part of renal tubule play important role in it. PCT, that's the first spiral structure present next to the Bowman's capsule, helps in maximum reabsorption of the nutrients as well as water. About 70% of water, all the essential nutrients and the ions such as K plus, HCO3 minus and NaCl is reabsorbed in PCT. Alongside what happens, the toxic waste such as ammonia and H plus ion is given back to the filtrate. Okay, coming to the loop of Henle, descending limb of loop of Henle is permeable to water, but impermeable to electrolytes. So water absorption takes place in the descending limb of loop of Henle, and ascending limb of loop of Henle is impermeable to water but permeable to sodium chloride. So what happens here? The sodium chloride is reabsorbed, water stays inside the loop of Henle. So if you see, as the water comes out here, descending limb concentrates the filtrate or is a concentrating segment, and ascending limb, because water is present, solute comes out, the filtrate becomes what? It becomes dilute. So ascending limb is otherwise called as the diluting segment of the renal tubule, right? Now let's look at the DCT, that is the third part. Here the reabsorption of the salts such as NaCl and HCO3 minus takes place just like that of PCT. Alongside, water absorption can also take place, but it is conditional. That means if the body requires it, absorption of the water can take place under the influence of the hormones such as ADH. So that is known as conditional reabsorption. And what is being secreted here into the filtrate? H plus ion, which is toxic for the body, and K plus ion is secreted to the DCT. Now if you pay close attention here, HCO3 minus, why it is reabsorbed by the body both in PCT as well as in DCT. Look, HCO3 minus ion is the most important natural buffer and is required for maintaining the pH of the blood. That is why it needs to be reabsorbed. Next point, why K plus ion is secreted first, why K plus ion is absorbed first in the PCT and secreted into the filtrate in the DCT. Look, K plus ion is another important ion which is required for the muscle contraction, also the nerve impulses, okay, acting of the nerves. So first, whatever K plus is lost in the filtrate, that is reabsorbed. But excess of K plus ion will also be harmful for the neurons as well as muscles. So excess of K plus ion which is present, that is given back to the filtrate in the last step in the DCT. Clear about these 2 points? They are very important concepts. Next, come to the collecting duct. What's happening here? Collecting duct, also water is reabsorbed. It is a main segment which is responsible for concentrating the urine. Some of the urea also diffuses out from the collecting tube, and that is mostly in the passive manner. Okay, this collecting tube, whatever water is reabsorbed, it takes place under the influence of the hormone ADH. Okay, let us now look at these like function of renal tubules in the tabular column. Let's now look at the summary table. The main site where maximum reabsorption takes place is PCT. It is because of the presence of special type of cells in them, that is the simple cuboidal brush border epithelial cells. Okay. Now 70 to 80% of water and 100% of nutrients are absorbed here. It also functions in maintaining the pH and ionic balance. Coming to the loop of Henle, the descending limb is highly permeable to water, while ascending limb is impermeable to water. So, descending limb is a concentrating limb and ascending limb is a diluting limb. The ascending limb is a site where the minimum reabsorption of the nutrients takes place. Okay. Next, come to DCT. DCT is the region where conditional reabsorption takes place, that is depending upon the body requirements under the influence of the hormones, reabsorption takes place. Its main role is fine tuning the urine, maintaining the ionic balance and acid base balance. Coming to collecting duct, its main function is urine finalization or maintaining the final concentration of the urine. The reabsorption of the water that takes place in collecting duct, that is mostly under the influence of the hormone ADH, which results in formation of aquaporins or pores that help in releasing water and hence concentrating the urine. So the final concentration of the urine is achieved in the collecting duct. Some amount of urea is also reabsorbed in collecting duct, which helps in maintaining the medullary osmolarity, and the reabsorption of the urea is mostly passive in nature. That is, because concentration of urea is more in collecting duct, small amount of it diffuses out into the interstitium. Okay, so that's all about the summary table. Now, as we have completed the conceptual part, let's now quickly see the important PYQ liners. Normal GFR, it is 125 milliliter per minute or 180 liter of filtrate per day. But what's the urine output? 1.5 milliliter per day. Okay. Now, ultrafiltration occurs due to glomerular blood pressure difference. Okay. Now, then what is the net filtration pressure? Net filtration pressure is 10 millimeter of Hg. What does filtration membrane consist of? It consists of 3 layers: capillary endothelium, which has fenestrations in them; basement membrane, which is non-porous; and podocyte epithelium present in the Bowman's capsule that has filtration slits or slit pores. So question can come: slit pores are present in podocyte epithelium or the inner layer of the Bowman's capsule. Okay. Then where are the fenestrations present? Fenestrations are present in the capillary endothelium. Now, the afferent and efferent arteriole, which has a wider diameter. Afferent arteriole has a wider diameter than the efferent arteriole. Look at the table here. Site of maximum reabsorption, PCT. Site of minimum reabsorption, ascending limb. Water permeable segment, descending limb. Water impermeable segment, ascending limb. Very, very important. Function of DCT, maintaining the ionic balance. Now, conditional reabsorption takes place in, answer would be again DCT. ADH acts on collecting duct. Okay. So these are the important PYQ liners. Practice them well. Okay. Now, this is the final urine pathway, like blood, it goes to glomerulus, where ultrafiltration takes place, then PCT, loop of Henle, DCT, collecting duct, and finally urine is released through the urethra. Okay. Thank you everybody. If you enjoyed the concept today, please hit the like button, subscribe to my channel, and share with your friends so that they can also get value from the lecture. If you have any doubts, please feel free to contact me. I will be very happy to answer. Thank you everyone. Have a very good day.